Hello, everyone, and welcome to today's webinar titled Artificial Intelligence and Multi-Omics in Perinatal Period. This webinar is part of the 2022 Microbiology Virtual Week event. And I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. So let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees in our new live chat feature during the presentation. You can find that live chat located on the right of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. If you have any trouble seeing or hearing this presentation, simply click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located in the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Josef Neu. For a complete biography on our speakers, please visit the Biography tab at the left of your screen. Dr. Neu, welcome. You may now begin your presentation.
I'd like to thank the organizers for inviting me to give this lecture. The title of this lecture is Artificial Intelligence and Multiomics in the Perinatal Period. And the agenda for the next 40 or so minutes is as follows. I'm going to give a brief background on artificial intelligence and machine learning. I'll give a brief background on multiomics, and then I'll talk about how we can mesh artificial intelligence with multiomics in diagnostics and therapeutics in maternal fetal medicine and in neonatology. And then I will just touch upon where we are going in the future. Let me start with this slide. Many years ago in the early 1940s, a very famous author by the name of Isaac Asimov wrote a series of books called I, Robot. And this series of books depicted robots with intelligence who one could actually communicate with. And at that time, certainly this was not possible in reality, but it was something that was just in the mind of the readers and also Isaac Asimov. So it was just fiction at the time. In the 1940s also, in the early 40s, there was a major war going on, World War II, and the German military had this code called the Enigma code, which carried important information that was critical for moving the war forward for the German army. But this person here on the bottom right, this is Dr. Alan Turing, was able to use some mathematics developed by some Polish mathematicians to break this Enigma code. And he is now known as the father of the modern computer. And one of his quotes was, "I propose to consider the question, can machines think?" Well, that was the 1940s. And we've come a long way since the 1940s. In 2022, a device worn around our wrists carries exponentially more computing capability than devices that required large, cooled rooms in the 1950s. We now are having self-driving vehicles, facial and voice recognition, and the ability to connect classrooms worldwide, and also the integration of human cognition with the rapid processing of artificial intelligence. And these are all examples of these major advances that have taken place in these last few decades. In medicine, we've also made considerable progress. Now here, this slide shows 3 eras of medicine: the past, present, and future. So in the past, we relied largely on intuition medicine. So, for example, in my field, in neonatology, we think that Premature babies have immature immune systems. So we have to use antibiotics a lot in these babies. And in fact, antibiotics are given to almost every premature baby weighing less than 1,500 grams in most neonatal intensive care units in the world. But this is intuition based. We don't know if this is really the right thing to do. In the present, we are now undergoing some associative studies, epidemiologic studies, and also prospective randomized trials that are hopefully telling us whether this is a good practice or not. In the future, we hope to be able to use precision medicine. In other words, be able to get information from each individual patient and be able to tell whether that patient should be getting antibiotics or not. And this will be an algorithmic-based approach. So here are some of the different modes of artificial intelligence. We have predictive analytics, which rely on a technique called machine learning. We have text to speech and speech to text. We have image recognition. And at the bottom, we see additional forms of artificial intelligence, which are just in the very early stages of being applied to medicine. In terms of publications related to medicine, when we talk about multi-omics and artificial intelligence, this is a slide that shows a logarithmic scale and the increase in various publications in these areas over the past 12 or so years. So marked increase in interest and publications in these areas. Now here's a picture of a type of a baby that I take care of in the neonatal intensive care unit. And this is a fertile area for artificial intelligence and multi-omic applications. For example, we want to be able to use predictive analytics, diagnostics, especially when coupled with multiomics, and also precision nutritional support. And I want to give you just some examples of these applications. Now here's a fellow who you might recognize. He is now 72 years old, but when I was growing up, this was little Stevie Wonder. He was a child prodigy and a musician, and he had some very famous songs, and he's wearing sunglasses. And the reason he's wearing sunglasses is not to be cool. The reason he's wearing sunglasses is because he is blind. He is blind because he has a disease called retinopathy of prematurity. Retinopathy of prematurity was first recognized in the late 1940s, and it increased suddenly in premature infants in the late 1940s. In the early 1950s, it was found to be associated with routine high oxygen exposure in closed incubators. It was initially called retrolental fibroplasia, but as more was learned, the name changed to retinopathy of prematurity. If you look closely at the picture at the top here, you see that this baby has very cloudy looking eyes. The pupils of the eyes look cloudy. The reason for that is because the retinas have detached, and that causes this cloudy looking appearance. And once the retinas detach from the backs of the eyes, the baby is likely to be blind. Now, we have some newer techniques utilizing artificial intelligence that help us in terms of diagnosis of this retinopathy of prematurity. We are able to look at pictures of the backs of the eyes, and rather than having different ophthalmologists have a different impression what the retina looks like, we now have algorithms that really tell us with a high degree of accuracy the stage of retinopathy of prematurity. And to look at the features in the backs of the eyes and the retina using these techniques is actually beginning to be more accurate than the diagnosis made by the ophthalmologist. We also have a terrible disease entity that we see in premature babies. I should call this a diagnosis rather than a disease. And this is called necrotizing enterocolitis. And here is a picture of a baby on the top left that has a distended abdomen. And you see a little bit of redness around the belly button. This baby had an x-ray taken, and the two arrows are pointing to features that are common in this disease. The bottom arrow points to air in the bowel wall, and the top arrow points to air in the liver. This baby continued to do worse and worse, and then was taken to surgery. And at the bottom left, you can see what this baby's intestine looked like at surgery. The arrow points to a necrotic piece of intestine, and that was resected, and the baby did well after the resection. However, about 20 to 30% of the babies with the surgical form of necrotizing enterocolitis end up dying of this disease. We also are beginning to recognize, as I mentioned before, that this is a diagnosis and not a discrete clinical entity. We do not have a good definition of this disease at this time, and we lump several different entities under the umbrella term of necrotizing enterocolitis. We have not made very much progress because of this. And we also have animal models that have been studied that really don't represent the disease that we see in humans. And we've had narrow focus on individual pathways rather than systems. And so we have not made very much progress in this with helping predict this diagnosis or preventing it. Here are just some of the imposters of what we are calling necrotizing enterocolitis. And so there are several different entities that fall under the umbrella here. Work that we did at the University of Florida just in the past couple of years involved looking at babies who have what we consider to be spontaneous intestinal perforation versus the true necrosis of the intestine. And on this slide, we see that using machine learning techniques, we have been working on trying to be able to utilize features of these two different entities that are called necrotizing enterocolitis to be able to tell whether this is true necrotizing enterocolitis versus spontaneous intestinal perforation before the surgeon actually goes in and takes a look. And so this slide shows the baby with a spontaneous intestinal perforation. And here again is the more classic form of necrotizing enterocolitis. The way we studied this was to take a large number of babies who had exploratory laparotomies, and we were able to take 40 patients who actually underwent the laparotomy and clearly showed surgical necrosis of the intestine versus the spontaneous intestinal perforation. And then using machine learning, we were able to find certain features, as seen at the bottom right here, certain features that fit more with one form of this problem versus the other form of the problem. And so we did two different kinds of machine learning models, and both of these machine learning models were able to separate the necrotizing enterocolitis, the true necrotizing enterocolitis, from the spontaneous intestinal perforation with a high degree of certainty. So what does this mean? This means that rather than going in and operating to try to find out what this baby actually has, the surgeon can predict with a high degree of accuracy whether this is a spontaneous intestinal perforation or necrotizing enterocolitis. For spontaneous intestinal perforation, it is easier to just put a little drain into the abdominal cavity rather than doing a full laparotomy, and that drain will then drain the peritoneal cavity, and the little hole will heal over, and the baby should do fine with just that procedure, relatively simple procedure, rather than opening the abdominal cavity completely. So this can be very helpful for the pediatric surgeon to discern these two different forms of intestinal injury. Next, I'd like to talk a little bit more about multiomics. And if we talk about omics, probably the first omic that we think about is the genome, or genomics. And genomics is something that has burgeoned over the past several decades. We now are able to evaluate our human genome very rapidly. We can, some of the laboratories that do human genomic sequencing are able to get DNA from an individual, and they're able to sequence this and provide the sequence in less than a day. So tremendous progress has been done, has been made in genomics. But there are other forms of omics. One of these forms is called the microbiome. And this has stemmed from work in genomics. Some of the technology developed in the Human Genome Project has been applied to microbial DNA sequencing and RNA sequencing. And we have several different forms of microbiome sequencing technologies. One of these is called 16S sequencing, which looks at a small portion of the microbial genome, a 1500 nucleobase region. And this is very helpful in telling us the types of microbes that are there, the relative abundance of microbes that are there, the diversity of the microbes. So it can be very helpful to some extent, but it doesn't tell us what the actual potential, the physiologic potential of those microbes are. It just tells us their relative abundance and who might be there. Whole genome sequencing, on the other hand, looks at the entire microbial genome, and the databases that are available for whole genome sequencing can tell us much more about what the actual potential of these microbes are, what they are doing in the niche that they are in. So we have applied microbiome sequencing to evaluate necrotizing enterocolitis and the types of microbes that are present prior to the development of this diagnosis. And here we see these two graphs at the top, and the graph at the left represents a time period with babies at 24, 25 to 36 weeks gestation, and the colors represent different phyla of bacteria. These are the control babies, and on the right, we see the necrotizing enterocolitis babies, the NEC babies. And one can see that the NEC babies over time from 24 to 35 weeks, if we sequence their microbiome, we see differences compared to the controls. The blue appears to increase. These are a certain phylum of bacteria called the proteobacteria. The yellow, the dark yellow, begins to decrease. And these are bacteria called the Bacteroidetes. We know that the proteobacteria are bacteria that have a lot of lipopolysaccharide. This is an agent in their cell walls that stimulates an inflammatory response. And so we are seeing more of these potentially pathogenic microbes develop prior to the diagnosis of this disease. And then the dark yellow represents the firmicutes, and the firmicutes, and these firmicutes produce certain types of short chain fatty acids that are known to be protective in the gastrointestinal tract. So this is telling us that there is an association between the types of microbes in the gastrointestinal tract of these babies before the development of the disease. However, this is an association, and it doesn't tell us whether this is actually causing the disease. So when studies like this find an association between two things, it does not mean that one thing caused the other to happen. So how can we get by this? And this is where multiomics comes in. And I really like to explain multiomics using this parable, and it's the parable of the 6 blind men of Hindustan. Each one of these blind men of Hindustan was asked to feel this structure. And one of the blind men went to the side of this structure and said, "This feels like a wall." The other blind man went to the back of the structure and said, "This feels like a rope." The other blind man went to the front of the animal, or front of the structure, and said, "This feels like a spear," and so forth. And all of these blind men came back, and they were dogmatic in what they felt. They didn't talk to one another. They didn't really collaborate. And this was problematic because they were not able to determine that this was truly an elephant. Had they talked to one another, collaborated, they would have been able to tell if this was an elephant. And this is where multi-omics comes in. And individually, each one of these omics, for example, the genome, can be very helpful. But you can have genomic mutations and genomic variations which are never truly expressed unless you have certain environmental conditions that come in and cause that genomic sequence to be expressed. And then you can see that in the transcriptome, in the proteome, in the metabolome. And so one can look at these different omics under different conditions over time and integrate them, and they can much more strongly identify patterns associated with disease and even causative of a disease or outcome. And once we have this can more readily lead to preventative or therapeutic targets. Here we see how different layers of these omics are placed on one another, and you can start with a phenotype, or you can start with a genome first techniques, and you put them together, and you find this pattern that can develop with these different nodes that we see, these circles and colors. And the larger they are, the larger these are in this mechanistic modeling, the greater is the interaction. And so using the systems biology integration of omics data, we can now much more readily look at these omics and be able to evaluate mechanisms of disease. So here is just one example. Inflammatory bowel disease, and this is a study that was published a few years ago to evaluate what happens when you look at single omics. With single omics, you can get secondary analyses. As I mentioned before, you will get associations. But if you put these omics together and you integrate them, you get primary mechanisms. So much better than when you look at a single omics approach in terms of defining mechanisms of the disease. So here we get back to this little premature baby, a 25-week gestation, intrauterine growth restricted female. This is telling us that this baby probably had poor nutrition in utero. And how do we nourish this baby? Well, in the past, using the paradigm that I mentioned before, we would say, well, we have these formulas available for premature babies that can be used, but all of the formulas are pretty much the same for premature babies. We have mother's milk, which could be used. This doesn't tell us very much about that particular baby and what is best for that particular baby. We're currently undergoing all kinds of studies to try to use evidence-based medicine to provide information on how to best nourish a baby like this. But in the future, we will be able to say this was an intrauterine growth restricted baby. This is a girl. This is a baby who had an infection at birth, and put all those features together and be able to tell what is the most appropriate nutrition for that particular baby. We are beginning to recognize that precision in nutrition is something that is very important, because we do not all respond the same to the same food intake. This is a study that was published in Nature Medicine a couple of years ago, where they looked at identical twins and gave these identical twins very similar diets. And then they looked at triglyceride, glucose responses, various nutritional responses, and they found that these twins had different responses to the same nutrients. But they found that there was a very high correlation to the type of microbes present in the gastrointestinal tract of these twins. And the responses were highly correlated to the types of microbes present in the gastrointestinal tract. So when it comes to nutrition, why personalized? Well, here we have two different types of premature babies, a 23-week preterm and a 29-week preterm. And the type of nutrition we need to give these babies is very different, because one size does not fit all. And we should be able to prevent some of the very adverse outcomes that we see in these babies by providing better nutrition from these babies for these babies early on. Now, we can provide different forms of formulas to these babies. We can provide baby's own mother's milk, or we can provide donor milk, banked donor milk to these babies. But we are beginning to recognize that baby's own mother's milk provides for the greatest precision nutrition. And on this slide, we see results of a study that were published in 2011, where 16 different 16 different mothers' milk samples were taken at 3 different time periods. And each one of these represents, each one of these S's represents a different mother. And we have these columns of colors that represent 1 week, 2 weeks, and 4 weeks after birth. And we see that the colors differ markedly from one mother to the other. So the composition, the microbial composition, which represents the colors that we see here, are very different from one mother to the next. So this is suggesting that the types of microbes that we find in mother's milk may be very much precision oriented for that particular baby. Why do I say that? In terms of personalization, the baby's own mother's milk over time is considered to be a highly personalized type of nutrition. And one of the reasons is seen here, something called the enteromammary immune system, where the mother and the baby, if they have close contact with one another, the baby's immune system may not be very well yet developed yet, but the mother's is. And so what happens is that any microbes that the baby is infected or colonized with will get into the mother's mouth by kissing the baby or being very close to the baby. And that will get into the mother's gastrointestinal tract, where her mucosal immune system sets up an immune response that then gets transmitted to the milk, and the baby will drink that mother's milk and take in some of the immunoglobulins that are specific for that particular microbe that the baby may be colonized by. So this is one of the rationales for seeing different types of microbes in different samples of milk. Again, this has not yet been fully tested in terms of rigorous studies, but it seems to be a very good, strong hypothesis that needs further testing. Work that we have done in our laboratories, two pictures here, Dr. Nicole Cacho, who was a postdoctoral fellow at the time, and Dr. Graziella Lorca at the bottom. She is a professor of microbiology at the University of Florida. We did some studies looking at the personalization of the microbiota of donor human milk with the mother's own milk. Well, donor human milk comes from lots of mothers, and it is pasteurized, and the microbes are killed by this pasteurization process. And that may actually be problematic in donor milk, that you are not providing microbes, and these microbes may actually be very helpful. So one of the things we did in this study that was published in Frontiers in Microbiology in 2017 was to take small amounts of the baby's own mother's milk, put that into donor milk, and allow that to incubate for different periods of time. And we found that the incubation period between 4 to 8 hours resulted in a refaunation of the donor milk, and a profile of microbes that look very much the same as the baby's own mother's milk. We also looked at the metabolites and found that the metabolites seen here in these principal component plots, that we were able to make the metabolites look much more like the baby's own mother's milk if they were provided with some of the small amount of the mother's milk into the donor milk, larger donor milk samples. So this is telling us that we may be able to do some personalization, and this is one of the pathways toward personalization of donor milk in the future. Now, how do we currently monitor nutrition in our premature babies? Well, one way is to use these growth curves as seen here on the chart. And we can see that we should be following these growth curves, but these dark lines represent growth faltering. This means that the baby is not growing the way the baby should. This is problematic, because once we see this kind of growth faltering, it is probably already late, and the kind of damage that this growth faltering can cause for this baby's entire lifetime can be problematic. We need good indicators that help prevent this. The growth curves are Gaussian distributions, and they are certainly not personalized. So we need to do something to be able to personalize nutrition. And this is something that we are starting to work on using artificial intelligence for prediction of growth failure. And here's a reference that was recently published in biomedical bioinformatics. And here we developed a model that integrates clinical and microbiome data. And the clinical and microbiome data was derived from a large British cohort and 3 cohorts in the United States. And we compared different models to get data on what would be the best clinical predictors of growth faltering. And we also found that information on the kind of taxonomy in the of microbes in the gastrointestinal tract may actually be helpful for us to predict this growth faltering. And so this told us that we need to move further and perhaps even take a multi-omic approach in being able to do this. So what kind of studies do we have planned in the future? Well, we'd like to be able to get timed infant samples from feces, saliva, blood, and urine, and then look at the microbiome, look at immune and inflammatory markers and metabolomics and proteomics, and put them together into this network analysis, and then be able to delineate the different diseases that might develop or adverse outcomes that might develop depending on the type of nutrition. We can then also do biomarker discovery, and then this could potentially lead to personalized therapeutic interventions. We still have a long ways to go with this, but I think that the tools are there. So here's a summary and where we are going in the future. The causal nature of single omic associations are not well understood, but they are amenable to analysis by newly developing technologies such as multiomics and artificial intelligence. Once these mechanisms are understood, we will better be able to provide predictive analytics and personalized nutrition for mothers and infants. And in order to accomplish this, we need to have teams of scientists. We need to have clinicians, basic scientists, engineers, and bioinformaticians working together and communicating and working as a team to be able to move forward and have positive accomplishments in these areas. Thank you for your attention.
And thank you, Dr. Neu, for that time today and your important research. And before we go, I want to thank our audience for joining us today as well. Questions submitted today and during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until September 6, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.